Go ahead and get started here. I'd like to welcome everyone to our first Innovation Grand Rounds for 2021. Happy New Year to all of you, and hope everyone had a restful break. It really is my pleasure to introduce Dr. Dan Llano today as our speaker. Dr. Llano completed his PhD here at the University of Illinois in molecular and integrative physiology, and then also completed his MD here as well in the University of Illinois College of Medicine. After that, he went on to complete his internship in internal medicine at Mass General Hospital, Harvard Medical School, as well as then a residency in neurology. Following that, he spent time at the Mass General and Brigham Women's Hospital, the Martino Center for Biomedical Imaging, which many people might be familiar with, and then a postdoc, and then finally back to the University of Illinois as an assistant professor in 2010, back in the Department of Molecular and Integrative Physiology, and also as a Carl neurologist. So you can see a theme here of clearly combining science with medicine in very interesting ways. And I'm sure that'll come out in Dr. Llano's presentation today as well. Dr. Llano has been very active as a research scientist, publishing over 70 publications in the areas of neurological and behavioral disorders, neurobiology, sensory processing, computational biology, imaging, and optogenetics. He has a number of awards. Some of the most notable is that in 2019 he was awarded the Presidential Early Career Award for Scientists and Engineers, which is an extremely high achievement. From the teaching aspect too, I was noticing from your CV that he's received the Excellence in Teaching recognition from our campus, and you say for every year, for every course that you have taught. I have not seen that before, but that's a wonderful testament to teaching and connections with students as well. Dr. Llano currently is an associate professor of molecular and integrative physiology, full-time faculty member at our Beckman Institute, and a cognitive and behavioral neurologist at Carle Health. He is a fellow member of our Carle, Illinois College of Medicine, and is recognized as the Benjamin R. and Eleanor W. Bullock and Edwin and Jeanne Bullock Goldberg Professorial Scholar. So quite a title there, but clearly is testament to your skills and achievements. So we're very much looking forward to your presentation today, Dr. Llano. Your title there is very interesting. We're all curious to hear about that. And I'll turn it over to you. So welcome and thank you.
Great. Well, thank you so much, Steve. It's really a pleasure to have been invited, and nice audience here. So I'm looking forward to the feedback here at the end. So yeah, the title of my talk is Sensory Perception, a Controlled Hallucination, Evidence from the Thalamocortical System. So the goals of my lab and the goals of this talk, I'll summarize now. So our work is about sensory physiology. We're interested in how the brain processes sound. The work that we do, the nuts and bolts of it is pretty basic science in terms of what we do on a day-to-day basis. But there is a longer-term translational goal that we do have in mind. The longer I've been doing this, I've been doing this now for over 20 years, it's pretty clear that perception is quite a fragile thing, and it's difficult to understand how it's controlled. And what we see clinically is that perception is often corrupted across really a number of disease states. So characteristically, you can see things like hallucinations and schizophrenia, and dementia with Lewy bodies and other dementing illnesses as well, delirium, just to name a few. And I think a good way of thinking about hallucinations is that they may in fact be a final common pathway of a sick brain, and that is the regulatory systems that are normally involved in controlling our perceptual processes. When they are not working, those perceptual processes become untethered, and people can develop false percepts or hallucinations. So our goal, again, the proximate goal, is to understand how perception is controlled, what the mechanisms are that control how we perceive the world. And my hypothesis is that once we understand those mechanisms, what I'll call the gating mechanisms of perception, I think that ultimately is what's lost during hallucinations, and hopefully we can pursue that down the line. Before we launch into the talk, I really would like to do a few acknowledgments so that I don't forget to do them later. So all the work I'm going to tell you about today was done by a postdoc in my lab, Bahar Ibrahim. Dr. Ibrahim's really been a special individual in the lab. He's done, as I mentioned, all the work, the experiments here, and many, many more that we don't have time to talk about. And these are very challenging multi-part experiments that he just does with ease, and couldn't present any of this work without his contribution to the lab. And then there are a couple others I'd like to just acknowledge, even though I don't know either one of them, Rafi Malak and Rafael Yusta. These are both visual neuroscientists whose ideas have really influenced my thinking about sensory perception and how it is regulated. Okay, so I think we can begin with what I'll call the dominant model of sensory processing. This is certainly what we tend to teach students when we're teaching them about sensory systems in the classroom. And that's this idea that there is a world that's out there full of objects. And if we're talking about the visual system, objects that we can see, and that our perception of those objects is sort of like a running movie that's playing on the back of our head. In the case of the visual system, that's where the visual cortex is. And that due to various factors like attentional factors, for example, maybe you get some distortions in this running movie, but ultimately there's some screen back here upon which we are playing or projecting some impulses from some subcortical center, the thalamus in this case. And although I think there is actually lots of evidence to support this type of model, I think it's quite incomplete. And I hope to make the case to you today that there's other ways of thinking about thalamocortical processing of sensory information that will complement this sort of movie projector model of sensory processing. Now, one of the problems with this model that I just presented is that it cannot explain a lot of what we perceive. For example, we perceive lots of things that are just simply not there. And this is a classical example, and we can spend hours showing multiple other examples like this, where in this particular case, most people will perceive a triangle that isn't there. What we are using to perceive that triangle are these little clues, these little hints, and those hints are likely calling up some internally stored representation of the many, many triangles that we've seen throughout our life prior to this. Now, I'm going to play you an audio clip, and I'm sorry, I'm going to stop sharing for just 1 second to be sure that I have the audio set up on this. So I'm going to play for you an audio clip, and the first clip I'm going to play for you, just want you to listen to it and see if you can understand what's being said. Okay, more than likely you weren't able to understand that. That was a clip that was basically most of the frequency information was removed. I'm going to play you the clean version of the clip, the unvocoded version of the clip.
A boy fell from the window.
And now I imagine most people would do much better at being able to understand that same version that I played for you at first. And so the idea that I'm trying to get across here is that with just a tiny amount of information, just some anchor points of what is being presented in this case to the ears, we do really a fantastic job of inferring what's out there rather than really processing the details of what's out there. And I think sensory perception is likely much more of an inferential calling up process rather than a moment-to-moment processing of the details process, at least in terms of our day-to-day life. Now, if we take a look at how sensory perception might be altered in things like hallucinatory states, we need to know, first of all, how various senses are represented on the cerebral cortex. Here's just an image on the left showing various parts of the cerebral cortex that are involved in sensory processing. So visual information goes back to the visual cortex, auditory information is represented in the auditory cortex and the temporal lobe, etc. And so if you take a person who is actively hallucinating, and these studies have been done, for example, here's an example of somebody having a visual hallucination while getting an fMRI scan. And what you can see is that the parts of the brain that are lighting up are the parts of the brain that would normally activate when this person is seeing real objects in the world. And that is, these are parts of the extended visual cortex, lingual gyrus, fusiform gyrus, etc. And although we don't know whether or not these patterns would be indistinguishable from somebody seeing whatever it is this person is hallucinating at this moment, the idea is that the same parts of the cortex that activate when we see something are also activating when we hallucinate something, when we are imagining something in our mind's eye. Same thing is true in the auditory system. Auditory cortical areas activate during auditory hallucinations. And there's multiple studies that have shown similar types of studies, suggesting that these cortical networks contain stored representations of objects in the world that we've experienced in the past, and that these stored representations are being activated abnormally in a hallucination, and might very well be activated when we are experiencing the world as we normally do during non-hallucinatory states. And so that, I think, brings us to a question: could hallucinations and sensory-driven perception share similar mechanisms? And is the difference between normal sensory perception and a sensory hallucination a difference of gating or control of that, of the cortical representation of whatever it is that's out there in the world? And there is some evidence to suggest that gating is impaired in patients who hallucinate. So there's a classical finding that's been known for decades. If you take patients with schizophrenia and looking for example, a biomarker of schizophrenia, one biomarker of schizophrenia is the response of these patients to paired stimuli. So if you take a normal individual and you play a click, a very short duration sound, and you measure their EEG activity, their brain activity on their scalp, what you will find is a very characteristic positive wave, this downgoing positive wave at about 50 milliseconds that represents the response likely happening in the auditory cortex. If I present another click very soon after that first click, that second click or that response to that second click is diminished. It's as if those gates have been closed. And that's a normal process that happens routinely. Now, if you do the same experiment with somebody with schizophrenia, here's the response to the first click, and then here's the response to the second click. You don't have the same level of gating. And this is, again, a finding that has been around for decades. We model this in animals. It's quite a robust finding, but it suggests that there is a problem with the ability to gate or otherwise regulate the cortical networks involved in sensory perception in patients who hallucinate. So that brings us to the brain. So the question is, what's happening in the brain during sensory perceptual processes, and what's the dominant model of sensory processing? So I think it's fair to say most sensory physiologists still adhere to what I'll call a serial hierarchical processing model in sensory systems. And that is, if you take some sensory receptor out in the periphery, so for example, a cell in the retina, retinal ganglion cell, it's going to have a receptive field, some area of space that it responds to, and you can link up those various receptive fields of various different sensory receptors, have them converge upon some next layer in the model, and you now create a neuron which is responsive to some new feature. And you can think of all sorts of creative ways to link up these various receptive fields to create more and more specific filters. And this process is intrinsically hierarchical and it intrinsically relies on serial processing. And eventually, at the highest levels of the hierarchy, presumably deep into the visual cortex, you find neurons that are responsive to behaviorally relevant objects. And I don't think it's a straw man to say that this is still the dominant model. Here's just some images of recent review articles from the visual system that all espouse this type of serial hierarchical model with receptive fields being shaped at each level. And again, this may be true at certain levels of sensory systems, but as I'll argue today, I think it's very likely not to be true at the thalamocortical level. And here's part of the reason why. There's an emerging body of literature similar to what I'm going to show you here. Here's an image from the cat visual cortex, imaged using voltage sensitive dye, paper that was presented here almost 20 years ago from the Grunwald group, where they use voltage sensitive dye to image both spontaneous and driven activity in the cat visual cortex. So here's just a pattern, a random snapshot of activity in the cat visual cortex at one moment in time. Certain areas are activated and certain areas are suppressed. And then at a later point in time, they showed the cat a particular kind of image, a grating, just a typical visual stimulus. And you can see here how similar the spontaneous activity appears to the evoked activity. And the experimenters commented that it's very peculiar that sensory stimuli appear to be basically calling upon patterns of activity in the cortex that may already exist. Now, there was another study using a very different system here. This is the somatosensory system. This is in a brain slice, so a very different approach altogether, a different imaging technique. Somatosensory slice from Rafa Yuste's lab, where they elicited cortical activity by stimulating thalamocortical afferents. All these little circles represent neurons in the somatosensory cortex. The dark circles are the ones that were activated by thalamic stimulation. Later on, they let this brain slice just oscillate on its own, just have its own spontaneous activity. And they saw patterns that look like this. All these green cells are all being activated spontaneously. And in fact, if you look at the overlap, they look pretty similar. Very similar populations of cells are being activated spontaneously as are being activated by what would be mimicking an external stimulus. And there's a number of papers now that have shown similar phenomena, and that is spontaneous activity resembles in sort of an uncomfortable way the patterns of activity that you can evoke with sensory stimulation, raising this idea that perhaps sensory stimulation is calling upon pre-existing patterns of activity of the cortex rather than simply projecting activity on an empty or a blank cortical sheet. So this raises some questions that we'd like to answer. So do sensory inputs gate what I'm going to call pre-wired cortical activity that is wired up based on our experience with the world, that we build up these representations, and then when we later on engage with the world, we're calling them up rather than actively encoding every detail. And if this is the case, what's the mechanism that's controlling this activity? Getting back to the idea of hallucinations as being a problem of cortical control, where might this gate be? And is there a way that we could potentially control that gate, fix the gate in people with hallucinations, etc.? Okay, so how do we approach this kind of problem? As a neuroscientist, there's all sorts of ways that one could study this problem. You can do a human psychophysical experiment, you can do animal psychophysics, you can study neural circuits, do molecular analysis, or something in between these levels. In our particular case, we've been focused on the neural circuits, and the main reasons are we have a pretty good idea as to where to look. We think the thalamocortical circuits are likely going to be a rich source of understanding the gating of cortical activity. And what needs to be worked out is exactly how these areas of the brain talk to each other. And that's what we're going to talk about today. Of course, we always thinking about one level up and one level down, and trying to incorporate molecular tools, and we're now doing some more behavior to try to understand how these circuits influence behavior. But most of what we'll do today is at the neural circuit level. And our work is focused on the auditory system. So I'll just briefly introduce the auditory system here. Information comes into the brain, auditory information comes in through the auditory nerve, gets processed through a series of auditory brainstem nuclei. From there, it gets projected to the inferior colliculus, a major auditory way station in the midbrain. From the inferior colliculus, there's projection to the auditory thalamus, often called the medial geniculate body, or MGB. And then from there up to the auditory cortex, where most models would speculate that's really where we our conscious perception of that information comes into play. Now, there's a brain region that's interposed between the thalamus and the cortex. It's quite poorly understood. It's known as the thalamic reticular nucleus. It's a thin sheet of GABAergic or inhibitory neurons. And as we'll talk about today, we think this region is quite important in controlling thalamic outflow. So we focus in just on this thalamocortical part of the circuit. We can see it a little bit more closely here what's happening. We have information coming up from the inferior colliculus. You'll hear me call it the IC throughout most of today's talk. And then from the thalamus, which again is the medial geniculate body in the auditory system, we have a projection up to layer 4 of the auditory cortex. And what is not so apparent from this figure is all of what we'll call the descending projections. And in fact, there are many more descending projections than there are ascending projections. There's a massive descending projection from the cortex, from layer 6 down to the thalamus. That's a positive or excitatory projection. And there's also projection involving the thalamic reticular nucleus, which is inhibitory onto thalamocortical cells. And then this thalamic reticular nucleus is getting an input from this cortical thalamic projection. So we have this complex arrangement of both excitatory and inhibitory projections onto this thalamic neuron, a system that looks quite suitable to act as a gate or a mechanism to control thalamocortical activation. And so that's really what we're going to be asking in the experiments that I'm going to tell you about, is how is cortical activation going to be gated or controlled. Okay, so here's what we did. And so our work is being done in mice. And as you'll see, part of the reason why we use mice for these experiments, they have quite good hearing, but also there's quite a few genetic models of mice that allows them to be quite useful to dissect the various components of the circuitry that we'll be talking about. So the first kind of mouse I'm going to talk about is one that expresses something called GCAMP6. GCAMP6 is a genetically encoded calcium indicator. And in these mice, GCAMP6 is expressed in all excitatory cells. So you can image these neurons as they activate because calcium rushes into the neurons and those neurons fluoresce. So we take a mouse and we play various types of sound, in this particular case, 5 kilohertz pure tone sound. And then we can image using something called a macroscope, the activity in the auditory cortex. And in fact, these signals are so strong that we can image them through the intact skull of these mice. And so if you do that and you look at the response in the auditory cortex, what you get is a pattern that looks like this. This is a very typical pattern. So it's a heat map showing activation of various parts of the auditory cortex, where to the left is rostral, or to the nose end of the animal, to the right is caudal, so to the back end of the animal, and then dorsal, which is up, and ventral, which is down. And then what we see is 3 areas of the brain activate in response to sound, something called the primary auditory cortex, A1, secondary auditory cortex A2, and the anterior auditory field. Now, this image that I'm showing you is not novel. Many people have shown that you get 3 or a handful of areas of the cortex that light up in this kind of paradigm. But what I'm showing you is an average. This is an average of 40 responses after 40 different tone presentations are given. What I think is much more instructive is to look at the individual responses. So these are the individual responses to the 40 different stimuli that were given. And when I first saw these data, it was really quite surprising. We're so used to looking at the averages, you don't appreciate just how much variability exists in these data. Sometimes the response in A1, for example, is quite strong. Sometimes it's not there at all. And there's no obvious pattern to this. And so it brings up a number of questions of, well, why is there so much variability in these responses? And can we characterize what kind of variability is going on? Well, the first thing we did is we just looked at the distribution of the magnitudes of the responses. And it looks like there were 2 clusters of responses, a handful of times where there was a response of some magnitude, and then a handful of times when there was essentially no response, where the change in fluorescence was, bordering 0. And we call these off events and on events. And in looking at this, to me, one of the first impressions I got was that, well, this looks like there's some kind of gating mechanism that's involved here. Sometimes the gate is open and one sees on events, and sometimes the gate is closed and then we see off events. And so what we attempted to do then is to try to understand this gating. What are the mechanisms that control whether or not this signal actually gets through to the cortex? And to do that, we turned to a different kind of preparation. Instead of using a whole animal in vivo preparation, which is useful for these kinds of experiments, but quite difficult to do real circuit dissection, we moved to a brain slice preparation. And a brain slice preparation is quite useful because you can very easily target particular components of a circuit. We can keep these brain slices alive in a in vitro setting for 8 to 12 hours sometimes, which allows us to do multiple different manipulations of the circuit that really wouldn't be feasible in vivo. And we discovered about 6 or 7 years ago that you can cut the mouse brain in such a way that you can actually retain connectivity between the inferior colliculus, which is the midbrain, the medial geniculate body, which is the thalamus, and then the auditory cortex here. And when you cut a section in just the right way, you can retain those connections between all of the relevant structures: the inferior colliculus, medial geniculate body, thalamic reticular nucleus, TRN, and auditory cortex. And I'm going to show you quite a few images today that look something like this. So just hopefully we can, keep all these abbreviations in mind during the experiments. So here's one of the first experiments we did in the slice. So here's the brain slice, a diagram of it on the left. And what we did is we electrically stimulated the inferior colliculus. Of course, we can't play sound in this particular configuration, right? This is a brain slice. So the next best thing is to electrically stimulate these ascending projections from the inferior colliculus. And then we can image the activation across the brain. And we can image in a number of different ways. We can use calcium imaging, like I mentioned before. You can use something called flavor protein autofluorescence imaging. We've used local field potentials to look at these responses, and really a number of ways you can characterize the responses. But here's a very typical image of what you will see in this brain slice when you electrically stimulate the inferior colliculus. You see a response in the MGB, TRN, and the auditory cortex. And again, this is an averaged image. The question is, well, what happens if you look at each individual stimulus? And here's what you get. In the inferior colliculus, we can see all of the individual responses, just the time course of each individual response. And it's not particularly surprising that every time you stimulate the inferior colliculus and you're imaging right in that same area, you get a nice, robust response. When we looked into the auditory thalamus, or the MGB, again, every time we stimulate, we get a response, not quite as robust, but it's quite consistent. But then looking in the auditory cortex, we see something like this. And we've seen this now very consistently. And that is, you get these quite strong responses, but then for periods of time, you get these absent responses, what I'm going to call an off response. And this really perplexed us for a while. There really isn't a good model to understand why sometimes at a population level, you sometimes see this quite strong, robust response, and other times see nothing. The behavior here is stochastic. And again, we don't have a model to understand why this should be the case. And so we began to try to investigate what's going on, what's controlling these kinds of responses. And one idea that had been that had crossed our minds is that, well, maybe we get these up and down responses because there's ongoing oscillations in the auditory cortex. You might be aware there's something called up and down states in the cortex, and under certain conditions, these up states and down states can be seen in brain slices, and maybe when our stimulus happens to catch an up state, that's when you get a response. And when you happen to be in a down state, you don't get a response. So maybe there's just some intrinsic oscillation that is controlling this. And so we look for that by going back to the slice, electrically stimulating, imaging the slice, but now recording from neurons in the auditory cortex to see if we can find these up and down states. So here's what this looks like. Here's a trace in the auditory cortex, an imaging trace. So we see 12 different stimuli are being given. 3 of the times we got good responses. The other 9 times there was no response. And when we look for these intrinsic oscillations, in fact, what we found was that every time there was a response in the auditory cortex, there was an electrical response as well. This is a whole cell patch clamp, so it's an intracellular recording. And when we look more closely at these responses, these in fact are up states. So it's actually not the case that there was intrinsic up and down states that we happened to catch a ride on, so to speak. It was the other way around. That is, when we stimulated the auditory cortex by initially stimulating the inferior colliculus, we were triggering up states. So we're getting up states in response to the cortex. And again, up states have been well characterized. These are network level phenomena that are thought to be stereotyped activations of a whole column of the cortex. And that appears to be what we're inducing by stimulating the inferior colliculus. But this question then came up as to, well, that's fine, we're inducing up states. Why don't we see anything in between? Why do we sometimes not get a response? And one very simple hypothesis, very easy to test, is, well, maybe there's inhibition during these off responses, and that's why we're not getting responses. So how do you look for inhibition? You block inhibition by using a drug. So we went ahead and we redid these experiments, stimulating the inferior colliculus, imaging the 3 structures as mentioned before. Here's a control case, as we've seen now before several times. Inferior colliculus responds regularly, MGB responds regularly. Auditory cortex has these off responses. We expose the whole slice to blockade of GABA, that's an inhibitory neurotransmitter. And what we found were that the responses in the auditory cortex came back. They weren't any bigger. Overall, there was no change in the size of the responses, but the likelihood of inducing a response came back in response to GABA, and then that went away with the wash. And here's just the summary data here. So it does appear that inhibition is responsible for the off responses. And the next question came up is, well, where is this inhibition? Is it in the cortex? We didn't see it with the current clamp recordings, the regular voltage recordings, but sometimes there could be inhibition that could be hidden with current clamp, but you can see it with voltage clamp when you record the currents. And so we did that. So we went back to the slice, stimulated the inferior colliculus, imaged the slice, and now recorded from the auditory cortex in voltage clamp. We hypothesized that during the off responses, we would see this burst of inhibition. And we did the same thing in the MGB. And in fact, we found the exact opposite. During the off responses, we found nothing. During the on responses, we found inhibition. And in fact, the pattern of inhibition resembles exactly the kind of inhibition you see during upstates. Again, during upstates, there are both excitatory and inhibitory inputs that are being activated. So that was consistent with our previous data. We then looked into the thalamus, the MGB, and here we did see lots of inhibition. We saw lots of inhibition every single time we stimulated the inferior colliculus. We quantified the inhibition. And what we found was that there was more inhibition during the off events than during the on events. There was no change in the net amount of excitation in these slices, indicating to us that perhaps the MGB, the thalamus, may be the site of inhibition, which is responsible for the gating that we're seeing at the level of the auditory cortex. Well, one way to test that idea is to go back to these experiments where we block GABA with this drug called gabazine. But the trick here is to apply, so we're again stimulating and imaging as we were doing before, is now to apply gabazine in very selected locations. And I'll just mention that Bahar Ibrahim, who did all these experiments, came up with a way of doing this. It's actually quite challenging to apply a pharmacologic agent in a slice that in an area large enough to silence a whole brain region, but without silencing the whole slice. And he came up with a very nice way of doing this and did all the appropriate controls. So we've been able to silence the auditory cortex or silence the MGB specifically. And here's what we found. Under control conditions, this is imaging in the auditory cortex. Again, as expected, we see these off responses interspersed with on responses. When we silence inhibition in the MGB, those responses come back as we expected. And when we silence inhibition in the auditory cortex, it didn't have any impact. We again saw the off responses, and the wash in both cases took us back to our baseline state. And here's the overall data here. So these data are now pointing quite clearly to the thalamus as a site for the control of activation of the cortex, as a site for the gate of activation of the cortex, and that inhibition appears to play a key role here. So then the question is, well, where does this inhibition come from? So here's a diagram of the circuit of the projection from the inferior colliculus to the MGB to the auditory cortex. And as you can see, there are two main sources of inhibition. There is an ascending inhibition source from the inferior colliculus, and then there is a descending inhibition source from the TRN. And they interact. And the question is, well, which of these two appears to be responsible for the inhibition that we're seeing to control gating? And so we attempted to silence each of these projections individually. So how does one do this kind of silencing? There's a number of ways of doing it, and we tried several ways. So one way is using optogenetics. So optogenetics, this is a technique where one can express a light sensitive channel in a particular tissue of the brain, and then use light to either activate or inactivate groups of neurons that are expressing that channel. So what we did is we expressed or we injected a virus, so as an adeno-associated virus that induced the production of halorhodopsin only in GABAergic neurons. So we injected it in a what's called a Cre recombinase expressing mouse. So the mouse is expressing Cre recombinase in GABAergic cells. And that way we could only silence the GABAergic cells in the structure of interest, in this case, the inferior colliculus. So, and this image is just showing you it's a fluorescence micrograph showing you the distribution of the expression of this virus, which is in the IC, and its GABAergic projections to the MGB. So then we went back, we electrically stimulated the inferior colliculus, and then you can use green light to selectively silence all of the GABAergic neurons here. So you can wipe out GABA selectively in one structure, in this case using light. Because we were using light for these studies, we couldn't easily image the auditory cortex, so we use a local field potential recording in the auditory cortex, which works just fine, and we found no impact. We didn't find any change in the percentage of off responses using this paradigm. Now, there's other ways of doing this experiment. We also tried what's called a chemogenetic approach, which showed very similar responses. Okay, well, what about this projection from the TRN back to the MGB? So we also attempted to silence this, but we first looked at the projection from the cortex down to the MGB. That cortical projection, again, has a branch to the TRN. And so we attempted to silence it. We think that might really be the gate, so to speak. And so we use a very similar approach. We used a mouse that expresses Cre recombinase only in layer 6 neurons. It's known as an NTSR1, neurotensin receptor 1 mouse. And one can use a virus to express halorhodopsin selectively in these corticothalamic cells in layer 6. That's what's being shown here, with the fluorescent signal representing where the virus expressed. We can then electrically stimulate the inferior colliculus, as we typically do, use a green light to silence the corticothalamic projections. And what we found is that and then use a local field potential to record the responses. And what we found is that in silencing these corticothalamic fibers, we were able to recover the missing responses, suggesting that the corticothalamic fibers were responsible for creating the missing responses. Again, responsible for the gating that we've been talking about. And so now the question is, the corticothalamics are involved. The corticothalamics, we know, branch and innervate the TRN. Is the TRN itself involved? Is that the final inhibitory pathway that's really shutting down the MGB? So for that, we did a pretty simple experiment, no fancy optogenetics this time. We simply went in and we silenced the TRN with a drug. So NBQX is a glutamate receptor blocker. And if you picospritz it in the TRN, you actually aren't silencing the TRN itself. What you're doing is you're blocking any of the excitatory inputs from getting into the TRN and activating it. So you're basically making it inert. It's not getting any input. And what Baher found when he did these experiments was that if you look at the middle trace here and you compare that to either the top or the bottom, and that silencing the inputs to the TRN was what allowed us to recover the responses at the level of the cortex. These are all cortical traces that we're looking at here in the control condition and with the wash. And you can see the summary data here was quite robust. So these data, in our opinion, are quite squarely pointing at the TRN as the gate. Now, finally, the last couple of things I want to show you guys is there's a few pieces of the puzzle that didn't really quite make much sense to us. One was that if there's inhibition coming from the TRN, why do we still see quite strong MGB responses, even when there is no cortical response? And so what Baher did is he went back to the brain slice, and instead of imaging the whole slice, he now just imaged the MGB using a different kind of calcium indicator. It's a dye-based indicator to look at the responses of MGB neurons themselves while he was recording field potentials in the auditory cortex. And here's just a view of what this looks like. Hopefully you guys can appreciate this movie. This is just the medial geniculate body here. These are individual cells that are activating in response to electrical stimulation of the inferior colliculus. And maybe you can appreciate there are waves of activity that are occurring here. And what Baher did is he looked at the synchrony. How similar in time are these neurons being activated? And if you just take all of the neurons in a given experiment and you just look at their waveforms over time, you see something like this. This is the overlay of dozens of waveforms. And although it looks like it's kind of a mess here, there is this sense that there is some peak that's seen during the response. And if you were to compare that during on responses, meaning on in the cortex, to off responses, you can see here there really is no order during the off responses. So you're seeing MGB responses, but they're desynchronized. And he quantified the variability here and found that the variability was much higher during the off responses. So the MGB appears to be responding during on responses and off responses. It's just not responding in the same way. It's much more synchronized during the on responses, suggesting that the TRN is desynchronizing the MGB. And the last thing that we looked at is, well, okay, so we think we have a good handle as to what the mechanism of the gating is. It's still not clear why the gate is sometimes open and sometimes closed. It looks quite random to us, and this is a brain slice. We're not really, there's nothing happening in the slice. What's responsible for these for this variability? And so what Baher did is he went back to do the experiment stimulating in the inferior colliculus, imaging the whole slice, but now he's recording in layer 6. And instead of looking at the induced activity, what he looked at is the activity prior to the stimulus. And the idea is that, well, maybe there's something happening before the stimulus ever comes that makes the system more prone to respond, right? Is there something about the dynamics of the system that makes it more responsive sometimes and less responsive other times? And so he measured a period of time before each stimulus, about 1 second of time before. He did multiple different windows. And then we ask, is there something happening here that's interesting before the stimulus? And there's a couple of ways that we looked at this. One is we just took all of the information that one can extract in a 1 second waveform. So things like the power at the various frequencies, the phases at the various frequencies, and we put them into a classifier, work with our computer science colleagues up at the U of I in Chicago, and ask the question, which of these features appear to be most important in predicting whether or not you get an on response or an off response. And what they found was that the most important features were some of the higher frequencies that we were recording in the 30 to 40 hertz range, which correlates with gamma range oscillations. Now, we also did the same kind of analysis, or I guess a different kind of analysis on the same data using what I would call a brute force approach, which is taking the power spectrum of each period of time prior to the stimulus, averaging those power spectra, and then asking, is there a difference between the two groups, between on versus off? And we found a very similar pattern. And that is at the higher frequencies in the gamma range of oscillation prior to a stimulus, there was a tendency to have higher power prior to the on responses. And there are data out there, human psychophysical data, suggesting that some of our perceptual abilities are better when there is preceding gamma before a stimulus. Okay, so to look at the overall data, so here is our circuit from the inferior colliculus to the MGB up to the cortex, with cortical thalamus coming back down, and our two sets of inhibitory projections onto the MGB. So first of all, we showed this novel phenomenon of a nonlinear stochastic response in the auditory cortex after midbrain stimulation. And it looks like these off responses are being driven by inhibition at the level of the thalamus, not inhibition, for example, at the level of the cortex. We think that it's feedback coming from cortical thalamics that are recruiting thalamic reticular neurons that are coming back to close the gate at the level of the thalamus. We think oscillatory behavior in between the stimuli plays some role in controlling whether or not you're going to get a response or whether or not that gate is going to be open. And that the spatial temporal pattern of activity in the MGB is likely what is being controlled by the gate. So the language of the gate isn't so much to inhibit thalamic activity, but to change its pattern. And that will determine what gets through to the cortex. Okay, so if we go back to the original problem that I posed between this idea of serial hierarchical processing and not being consistent with this idea that you can get these on-off stochastic responses in the cortex, what I would suggest is that this foundational principle in sensory neuroscience of serial hierarchical processing is clearly important, but it's very likely to be important as we learn cortical representations, as we engage with the world and new information is hitting our sensory receptors. But as we engage with certain objects and as we have learned what they are, a much more efficient system is to activate those pre-stored cortical representations and to only send new information to the cortex if there's something different, if there's something that's been tweaked that would require us to attend to those particular features. The gating mechanism here, what's going to determine what gets through and what doesn't, is likely coming from cortical thalamic projections. And we think that's involving the thalamic reticular nucleus. And what this suggests then is that if there's a problem with the thalamic reticular nucleus, one can get wayward or abnormal activations of the cortex, abnormal activations or representations, leading to false percepts or hallucinations. Now, then the circle all the way back to the beginning, well, that would suggest that you would find problems in the TRN in hallucinatory disease states. Now, the challenge here is that it's very hard to study the TRN in people. As far as I'm aware, it's not something that you can easily image using either structural or functional imaging. I just can't see it. It's quite small. Pathological studies don't always look at the TRN or even look at the thalamus for that matter. But there are ways that you can look at TRN function in living human beings. So one thing that the TRN is involved with is the formation of spindles. So spindles, you might be aware, are oscillations seen in EEG waveforms about 8 to 12 hertz that occur during stage 2 of sleep. They're thought to be related to interactions between the TRN and thalamocortical neurons. And we normally will spindle away during stage 2 sleep. And when this has been looked at in patients with schizophrenia, they have a significantly diminished frequency of having sleep spindles, suggesting the presence of some pathology at the level of the TRN. In dementia with Lewy bodies, what's interesting about this particular disorder is that there is a loss of cholinergic function. So in many dementing illnesses, there is a loss of cholinergic function, but in DLB, it's more prominent than most other dementing illnesses. And when cholinergic input, specifically what's called alpha-7 nicotinic, particular subclass of cholinergic inputs, have been looked at the level of the TRN, what's been found is that it is quite diminished, meaning this level of cholinergic control on the TRN has been lost in these patients, and that might be responsible for their propensity to develop hallucinations. To my knowledge, nobody's looked at sleep spindles in Lewy body patients or in any other dementing illness. I think that might be a very interesting study to do at some point in the future. So I would suggest that targeting the TRN, or at the very least understanding the thalamic reticular nucleus during these disease states, may end up being a very fruitful way to try to control them. in a targeted way. And there's a rich pharmacology on these neurons that could be potentially targeted through various drugs. Okay, so I'd like to acknowledge more fully the people who did the work. As I mentioned, Bahar Ibrahim did all of the slice experiments that I showed you. Jeff Brown did much of the analysis. Gosha Yudinsev, who was a former PhD student in my lab, did some of the in vivo experiments that we're now repeating in awake animals. And Gong Zhao, a very talented grad student in my lab, is now working on this project. I had some great collaborators at UIC who helped with the classifier building, and Matt Banks and his grad student Caitlin Murphy helped out with some control experiments. So thank you very much for your attention, and I'd be happy to take any questions that are out there.
Wonderful. Thank you so much, Dr. Llano. That was really interesting. I think the chat has actually filled with questions. So I'm happy to maybe start from the beginning and read some of those for you to address, and maybe you can take a look at those too. So that first
Sure.
one was, yeah.
I mean, I'm happy just to read them off. It's a question here about interoception. In terms of internal organ activity in health and disease. That's pretty interesting. So interoception, which is really a function of the autonomic nervous system to process internal states of our various organs, generally speaking, does not come to conscious awareness. So the mechanisms that I'm really referring to here are mechanisms that likely determine whether or not we're, aware of whatever, a sound, a picture, a scene, not so much our internal states. And on top of that, the thalamocortical representations of internal organ states are just not well understood. So yeah, I don't have a good way to answer whether or not any of this is relevant for autonomic sensing, but it's a very interesting question. Okay, so how might the noise in our society and lives alter and affect our future perceptions? Well, so probably the most direct way that I could answer that. So, if you're referring to noise as a toxin, as a way to damage our sensory systems, and so to be very literal about it, loud noise, for example, will damage our hearing. And in general, loss of peripheral sensory function will release these stored representations. So if you go through any disease state that involves loss of sensory input, so whether it be loss of hearing, leading to things like tinnitus, loss of vision, leading to things like Charles Bonnet, hallucinations, loss of sensory input, leading to phantom limb phenomena. The loss of sensory input appears to untether these cortical systems. It's a very interesting area of research. And what we know is that if you lose any sensory system, there's very significant reorganization that occurs in the brain that very likely creates a state where these cortical representations are just more easy to elicit. So yes, absolutely, there's a relationship between what's happening with the external environment and the likelihood of developing abnormal, bothersome, unwanted percepts.
So I guess a follow-up question on that is tinnitus essentially an auditory hallucination because the sensory inputs are lacking?
Right. So absolutely it is. So, but what's interesting about tinnitus is that the hallucination is quite simple in its form, right? So auditory hallucinations in schizophrenia are complicated. They're voices, familiar voices, running negative commentary about your life. These are almost certainly cortically generated representations that are complicated and involve emotions and whatnot. Tinnitus typically is buzzing, high-pitched squeals, rumbling sounds, things that are very likely subcortically generated, but have been able to make their way up to the cortex and be perceived, likely through some sort of disruption at the level of thalamocortical systems. So, and there's lots of evidence to suggest that there's changes along the whole neuroaxis in tinnitus, both at lower levels in the central auditory system, for example, the cochlear nucleus, and higher levels like the thalamus and the cortex. So yes, it's a hallucination, but it has a very different generator almost certainly than something like speech hallucinations in schizophrenia. Okay, serial hierarchical processing resembles convolutional deep neural nets. Can we attribute hallucinations to our AI algorithms at times? I'm probably not the best one to answer this, but I am aware of work that people have done using deep neural nets and basically getting them to have spontaneous activity that some people have ventured to call hallucinations. That's about as far as I'm willing to go, but sure. I mean, deep neural nets were put together based on the way our sensory systems are organized, and so maybe you'll find similar pathologies in both cases. Okay, how is gate pre-wiring encoded? Is epigenetics a possible mechanism? Oh, that's interesting. So when I say pre-wiring, what I really mean is learning. As we engage with our environment and we experience new things, even trivial new things, a new face or a new facial expression in an old face, they're going to change our cortical representations. There's 40 years of data showing that as you take a laboratory animal and you change their environment and you measure and remeasure their receptive field properties, they are highly mutable, highly changeable. And so, is there a question out there? Okay. So I'll just go on. So so cortical maps, for example, maps for sound frequency representation or visual location, etc. These things are highly changeable. And so what you're getting at is, well, what's the mechanism of those changes? Could there be epigenetic changes that occur there? Absolutely. I mean, this is still a very active area of neuroscience research, what it is that changes the receptive fields of neurons in the cortex. And so, yeah, I think anything is on the table mechanistically, but that's ultimately what I mean by pre-wiring this plasticity in these cortical networks.
I'm not sure if Lee's trying to ask a question here or not. There's definitely some scrambled text here.
All right.
I'll go ahead and mute him.
Sure.
Sorry.
Okay, question from Karina. Is it possible to simulate or stimulate some senses through others that are more available in some situations? So yeah, I mean, there's been a lot of work done to try to replace one sensory modality with another. Certainly, if somebody has lost one sensory modality, there is a large area of cortex which has now become available, so to speak. And the younger you are, the more you can wire, for example, non-auditory information into the auditory cortex. There's a very rich literature in animal research. Famous scientist Mrgan Kassur at MIT did pioneering work 30 years ago showing that you can cross-wire auditory inputs into the visual cortex and visual inputs into the auditory cortex. That's very much dependent on how old the animal is and the types of experience that they have. Now, in a human, it's much more challenging to do that. But there are examples where people have been able to get one modality of information in through a very atypical way. And the example that I'm thinking of is, and I'm just staring to see if Chip Davies is on this call, because I know he's done some work in this area, is using tongue stimulators, where people have, the tongue is a very sensitive organ. And so you could potentially put some device on the tongue, have some inputs that can stimulate various parts of the tongue. And those inputs could be responsive to anything. I mean, they could be responsive to X-rays if that's what you want them to be responsive to. And then your tongue would then be responsive to X-rays. And eventually you would have a cortical representation of X-rays. So in theory, one could find any sensory epithelium, put in pattern stimuli, and human cortical networks are pretty good at eventually figuring out what is being encoded. In fact, we rely on that for things like cochlear implants, right? Cochlear implants are putting in a pretty gross signal, a 20-channel signal to mimic the 15,000 hair cells that are damaged. But because our cortical networks are pretty good at deciphering pattern activity as we experience and re-experience the world, we can decipher it pretty well. So anyway, I mean, there's lots of opportunities to replace one sensory modality with another. And the final thing I'll say about this is, and the reason this is likely possible is because the cerebral cortex, the auditory cortex, the visual cortex, the somatosensory cortex is very similar in these various parts of the brain. I mean, there are people who would argue that there's fundamental differences between cortical areas, but in terms of their core computational abilities, they're likely very similar. So if you can route auditory information to the visual cortex usually will eventually be able to figure out that information that's being sent to it. Okay, so Judith wants to know about taste and smell with COVID. So I am aware that there's been some studies looking at just the olfactory epithelium. I think the olfactory epithelium cells, the supporting cells, not so much the olfactory nerve cells themselves, but the supporting cells are lost in COVID, and that's why these patients lose smell in this disorder. I mean, smell is one of these things that gets lost in a lot of different disorders for all sorts of different reasons, and degenerative disorders is likely related to loss of olfactory cortex, entorhinal cortex. But in this particular case, I believe it's a peripheral epithelial problem due to the organism infecting those supporting cells. So Tracy wants to know, I'm not sure if I already answered that question with my discussion about sensory replacement, so I'll skip to the next one. Can internal state of the animal be tracked with the scene variations in perception? That's a great question that we are actively trying to answer. So one thing we don't know with our data, for example, we're seeing these up and down states or on and off states, is whether or not those on and off states occur because of the varying state of the animal. You can imagine how these experiments go. You have a laboratory animal and you're literally playing the same sound over and over again. And there are times that the animal will be attending to the sound. There's times that the animal might be attending to something else, ignoring the sound, and animal might be sleepy, etc. And so the internal state of that animal can make all the difference in explaining whether or not you get a response. It may be what controls the gait. And in fact, if the gating is occurring at the level of the thalamus is one of these brain structures that gets very significant input from arousal centers in the brainstem. So as the animal gets sleepy, the thalamus is going to shut down. So what we're hoping to do is to redo these experiments, the in vivo experiments, in animals where we can monitor their level of arousal. And there's a number of ways of doing that. In the old days, what people typically did is they put an EEG electrode into the brain or into the skull while these experiments were going on. But a less invasive way has become quite commonly used. And in fact, we're setting up to use it in our lab, which is just to monitor the pupillary diameter. You could just set a camera in front of the pupil, and people have now shown really nice data that as the animal becomes more aroused, as measured using gold standard techniques like EEG, the pupil dilates and the pupil constricts when they're less aroused. And so one can compare the likelihood of getting, for example, a cortical response when the animal is more aroused versus when they're less aroused. And this is really gonna, I think, change things. I can remember when I was a graduate student 20 plus years ago doing similar experiments where you would see variable responses sometimes in the brain, and we would wonder, is this animal getting sleepy? What's going on? Why are we getting this variability? And back then we didn't have the ability to do this type of monitoring. So yeah, we really hope to be able to answer that question in the lab down the line. Okay, so Dr. Bopart asks about accommodation in brain slices. Yeah, so the question here is, does accommodation in the brain slices, does it have anything to do with our accommodating repeated sensory stimuli over time? We don't know yet. So I would argue that what we're seeing in the brain slice is probably not accommodation, only because we aren't seeing a continuous drop off in the signal strength over time. The presence or the absence of a response is behaving much more like a stochastic response than just a drop off. But this issue of accommodation is an interesting one to us. As you repeatedly present a stimulus to an animal in a behavioral experiment, you have to be aware of the fact that the animal is going to once the animal realizes that there's no meaning to that stimulus, it's going to ignore it. And so tracking things like the animal's attentiveness, what the meaning is of that stimulus to an animal is going to be quite important. This is an area that we're trying to move into to be able to alter the behavioral contingencies so that certain stimuli are going to be, rewarded, and to get an animal to attend to them. So yeah, I think it is an important factor that isn't always paid attention to with these kinds of experiments. Yeah, so Ben has a question: does increasing stimulus intensity increase the magnitude of the cortical responses, reliability, or both? Yeah, this is a great question that I didn't talk about here, but in fact, the on-off responses that we're measuring are threshold responses. This is what we see at low stimulus amplitudes. If you increase the stimulus amplitude, what you get is basically all on responses, and they're all about the same amplitude that we were seeing at the lower stimulus amplitudes. And this should kind of jive with our everyday experience. At low stimulus amplitudes, at thresholds, there might be a stimulus. Maybe you'll hear it, maybe you won't hear it. Could depend on a lot of different factors: arousal, competing sounds, if you're attending to that particular category of sound. But if you're presenting a loud sound, then you're going to hear it every single time. So yeah, this idea of a gate being open or a gate being closed, it's easiest to investigate that at threshold stimulus levels. And so that's what we shoot for. And that's the reason we're seeing this particular phenomenon. Okay, Margarita has a question. The individual variability is large despite using mice from the same genetic background. Is the problem related to the age of the mice or related to the time when the experiments are conducted, circadian effects? Those are really fantastic questions that we do have to keep in mind. So yes, the mice that we use in any given type of experiment are from the same genetic line. For example, the GCAMP mice are all C57 black J mice that are virtually identical, yet there is variability in the responsiveness. And our job as experimenters, obviously, is to try to diminish that variability so that we can understand our data. And so the question is, what are the factors that produce that kind of variability? And there's tons, right? I mean, there's tons that are even just the way that you do an experiment. So in our case, we're doing brain slice experiments. And even though Baher has done, God knows how many experiments like this, and he does them the same way every time, there's going to be a little bit of variability in how much of a particular pathway is maintained. There's going to be variability in exactly where the electrode is placed in the tissue and how much time is spent between cutting the slice and putting the slice, in the chamber, etc. But you have questions here about age and circadian rhythm. So these are very important points. So these slices that we cut are in animals that are quite young. They're at about P14 to P18 of age. That's postnatal day 14 to 18. So they're immature mice. And the reason we use such young mice is so that we can cut these slices. These slices are, compared to if you look at the kinds of slices that people use in their experiments, these are really complicated slices, probably the most complicated slice out there. And although we've tried doing this in older animals, it's just not viable. And so we are using younger animals, but within that age range of P14 to P18, absolutely there's going to be some variability in exactly how much we can retain in a given slice. And then diurnal variations. So yes, we try always to do our experiments at the same time, but there's going to be some variability there as well. So yeah, I mean, these are all important points that we try to factor in as we do these experiments. Okay, so Ben has the question about young versus adult. So yeah, we've tried cutting these slices in adults and just without much success. Now, in theory, one could do these experiments in an adult slice by injecting an optogenetic tracer into the inferior colliculus, letting it travel to the MGB, cutting a simpler brain slice, one that just contains thalamus to cortex and doesn't contain the projection from the inferior colliculus. That we can do in adult ages. And that's worth doing. We've never done that. And so we can't compare between young and old. I will say this, the systems that we're studying, the thalamocortical systems, cortical thalamic systems, are established very early in development, in prenatal development. So by the time that we're studying them, they're fairly well established. So I don't suspect that we're going to see major differences, but we just haven't looked yet. And again, there are technical reasons why we've just not been able to. So Howard wants to know, have you done experiments where you silence layer 6 auditory cortex? And did this restore the gating? So yeah, that was the NTSR1 experiment. So those were NTSR1, those are mice that express Cre recombinase and in layer 6 cells. These are NTSR1 cells. And we used halorhodopsin. Oh, I think you're asking if we did a retrograde tracer, would that restore the gating? So yeah, we silenced layer 6 NTSR1 cells, and that restored, and that eliminated the gating. And you're asking if we could restore the gating. I guess I'm not really understanding your question. What I can say is this, if you were to activate the layer 6 cells, would you restore the gating? We've not done that. That would be a very nice way to close the loop, right? If we could, instead of getting rid of layer 6, somehow hyperactivate layer 6, or hyperactivate the TRN and show that we got more gating, that's actually a really good idea. We've never done that, but it would be a really nice demonstration of the network. I would expect that you could shut down thalamocortical transmission if you were to enhance the responsiveness of layer 6 cells, but we just haven't done that yet. So Mohammed wants to know, if we block the noise in patients using a noise canceling, is this helpful for the patients? So, so again, noise exposure absolutely is a risk factor for hearing loss and for tinnitus. More recently, people have found that it's a risk factor for other things like Alzheimer's disease. And so you're asking, if you were to block that with noise canceling processor, would that be helpful? Well, so certainly in the case, so in the case of tinnitus, I think it's very clear, because virtually all patients with tinnitus have some degree of hearing loss, that if you were to protect their hearing, you would have less hearing loss and you would have less tinnitus. I think that's even though I don't think that's ever been studied, I'm happy to be corrected on that statement. I can't imagine any other outcome, that if you could protect people's hearing, they would be less likely to develop tinnitus. Now, the other active question, and I know this wasn't your question, but it seems to be one that people are more and more interested nowadays. What about other consequences of hearing loss, like cognitive consequences of hearing loss? There is an emerging field that has shown through primarily population studies that hearing loss is associated with more general cognitive decline, specifically that hearing loss is associated with Alzheimer's disease. And so it's an important question, because if it is, well, then boy, we should really be protecting people's hearing so that they don't get Alzheimer's disease. But that has never been established, meaning that you can do a study to protect people's hearing, and that would make people less likely to get AD. I think that study might be getting off the ground. There's a well-known scientist in this area, Frank Lin at Hopkins, who I recall where there were some plans to get a study like that off the ground, but I don't think that has been established yet. Okay, Howard has a question here. When I think of the TRN within the context of the talk, with the ascending cholinergic neuromodulation from the peripeduncular nucleus comes to mind. In the brain slice, this input would be removed. Do you think gating to the TRN might be highly conserved, but that the input to the TRN that controls the gating could arise from multiple sources? Yeah, I think these are the types of questions that I would really like to answer, and I think that they are highly answerable in the preparations that we're using. So the brain slice prep really makes it quite easy to get in and do the kinds of experiments that you're suggesting. So the basic premise of your question is that there's all these inputs to the TRN that we cut off to cut our brain slices. So what would be the impact of those inputs if they were still there, and could we manipulate them? And I mean, you can think of a dozen different experiments you can do to do this brain slice experiment, and to get in there and to either block or to agonize cholinergic receptors, or some subclass or some subclass of cholinergic receptors, and see how that affects the likelihood of having a response at the level of the cortex. We haven't talked at all about dopamine. Dopamine is usually the neurotransmitter that's invoked when we talk about things like hallucinations, but there are dopamine receptors in the thalamus, in the TRN, and whether or not they have any role in the transmission of information up or back down that system hasn't been looked at, but that would be a very straightforward experiment to do. Nowadays, in the old days, we would do these experiments by pico-spritzing the drugs onto the various structures, which can work well, but it doesn't necessarily give you the fine temporal information. But now we could potentially pre-label a particular dopamine pathway or cholinergic pathway with an optogenetic probe, and then you can, activate it with really high temporal precision, either just before or just after you stimulate the inferior colliculus, and see how that affects transmission. So yeah, Howard, those are awesome experiments that we should do, and hopefully we'll get around to doing them. Okay, Cindy has a question. Is there a link?
More of a comment, but yeah, there's I think one more question in the chat, and then I know Bill might have a question he wants to ask too. So
sure. Okay, so question about naive auditory networks have never been initiated. The tests of so the question is, what if we did these experiments in animals that have never heard anything, animals that were deaf from birth or acoustically deprived? How would that affect these systems? So we're very interested, absolutely, in our lab in looking at the impact of either acute hearing loss or auditory deprivation on these networks. Now, I would say that these particular experiments would be very challenging to do in an auditory deprivation paradigm. Number one, because of the variability in the brain slices, you would really need a large number of animals to, I think, get any kind of real confidence here if you saw a difference between the groups. Auditory deprivation is actually kind of challenging to do in rodents. What people more commonly do is they use some toxin to deafen the animal, either a chemical toxin or a noise toxin. Deprivation would involve people put earplugs in these animals and whatnot. And there's some technical issues in doing that effectively. But yeah, I think what we know is that when you eliminate hearing from an animal, its central auditory system changes very significantly. I would hypothesize that descending projections that control these gating mechanisms would be particularly susceptible to those changes, because most of the changes that occur in the deprived auditory system are done to basically increase the likelihood that some now weakened sensory stimulus is able to make it up the chain and to activate the cortex. So I would predict that you would have less gating in an animal that's been either deafened or deprived. But again, we haven't done those experiments, and I think they would be challenging to do, but I think those are the kinds of experiments I'd be very interested in getting involved with.
Great. Yeah, great set of questions. So I know, Bill, you might have a question you'd like to ask?
Oh, hi, I'm Bill Brewer, cognitive psychologist. We're about to move the discourse up a level here, I'm afraid. But
okay.
Your overall approach to perception seems to me strikingly right on the target, and it's really good that you have found ways to do it. Let me tell you how I come into this area. I became interested in the fact that philosophers of science were making psychological claims without looking at the evidence from cognitive psychology. And so I've written a series of papers to them or for in their journals, trying to make arguments like you made at the beginning about what that perception has to be some mixture of top-down and bottom-up processes. So what I'm about to show you an example, I guess, or so, is it should make you feel good, because in some ways if you had found totally different results other than the the nice top-down bottom-up approach, it would sort of invalidate. It wouldn't necessarily. I don't think I'd give up. But anyway, I'll show you an example of what goes on here. So if you think about the positivist philosophers, the first round of philosophy, they took perception to be an hierarchical input. They didn't talk about the physiology, but their basic model was that the world, you take photographs of the world in your mind, sort of, right? And that's what, and then they took that to be so true, they accepted that that raw perception has to be the basis for science. Then, there was a counterattack by Thomas Kuhn in the 60s that said, oh no, there are lots of examples where scientists are seeing things that aren't there because of their strong beliefs. And then unfortunately, that was a slippery slope. And so that was then taken by postmodern and other people to believe that maybe science isn't to be taken seriously because it's all top down. And of course, you can imagine where I'm going here. I've written papers saying, hey, folks, a position in between where we have possibility of top down and bottom up is obviously the way to go. And so to give you a concrete example of just an intuitive one, the original people, the astronomers are often the people who are at the edge of ability to see things visually. And so they have a lot of top down, bottom up examples. So after Galileo had the telescope and looked at the satellites around the planets, they turned their telescope to Saturn, and they drew pictures of various kind of satellites going around the planet, trying to make sense of what the input sensory data was. And they eventually, as the telescopes got better, somebody said, hey, it's not a bunch of moons around there, that's a ring. And suddenly everybody could see the ring. So other examples, well, it's a two-edged sword in the history of science. If you have top-down material, and it happens that nature is that way, you win big. So the people who observed Neptune, there was a mathematical calculation where Neptune had to be, and they found it immediately after being, given hints as to where to look. But of course, another group of people were unhappy about the fact that there was problems with the calculations for Mercury, and they went in and looked. So they believed there must be an inner planet between Mercury and the sun. And so they went looking for it in very difficult position because it'd be right close to the sun. And they found it and named it. Vulcan was its name. And you can see here the nice interplay between top down and bottom up things. And if that's to make sense of my historical examples, you really have to have something, there has to be a physiology down below that allows this to be true, right? And you are, of course, providing that physiology. So I'll show, it's a big, large issue. We should talk sometime and I'll shut up.
Absolutely.
You see,
yeah, no, that's great. I mean, I'd love to meet and chat about this more. If you look at the organization of sensory systems, one cannot help but believe that top-down projections have a major role in our sensory perception, just based on the numbers alone. Looking at, again, if you look at the thalamus, about at least threefold, maybe some have claimed tenfold greater number of top-down projections to the thalamus than there are bottom-up. So clearly, the thalamus is going to be strongly influenced by prior beliefs. And if you look at classical work that's been done in the auditory system, if you look at receptive fields of peripheral neurons going all the way down to the level of the cochlea, the most peripheral that you can go, you can really significantly change the response properties of those cochlear neurons based on cortical stimulation studies. So the system is really built, it looks like it's built upside down. But I think it's fair to say that prior beliefs, stored representations are going to have a massive effect on even the most peripheral of sensory receptors, and that seems to go along with the kinds of things that you're suggesting.
Yeah.
Well, wonderful, Dr. Llano. This really has been a highly engaging hour and a half, and I'm sure there's many other questions. We're unfortunately probably going to have to step away here, but I can only describe this as what a wonderful stimulus and what a great response we've had today. So thank you so much. I'm sure you're available for questions if people want to.
Absolutely. Yeah, shoot me an email or anything, no problem at all. I appreciate the opportunity. Had a great time.
Great. And thanks to everyone. So until we meet again, have a good day, and we'll see you again next time. Thanks, everyone.
Bye-bye.